So, good morning, students. So, in the last class, we studied about the important questions of that biomolecules. So, we discussed already the elucidation structure of glucose, and we wrote yesterday's class the Fischer structure, which is important for Thomas. And also from that Fischer structure, we need to write the Haber structure. Haber was a scientist who gave a arrangement of that what the cyclic structure is there. As I told you, those cyclic structure resembles the another organic molecule. Okay, so by comparing with that, you compare the glucose with the pyranose structure. Okay, so I'll explain, then you came to know. Okay, so initially I write Fischer structure of glucose. Fischer structure of glucose. Okay, so in that alpha D plus glucose is there, beta D plus glucose is there. I'll consider here that is 1, 2, 3, 4, 5, 6 atoms. Here one is, of course, we know that CH2OH, here it is CH, and the remaining are what? HNOH. So students, here this carbon, of course, this carbon is cyclized. Carbon number 1 and carbon number 5 is cyclized, and this is OH, H. This one is OH, H, H, OH, OH, H. Okay, so if it is like OH group is this side, it is alpha D plus glucose. Same structure. So I'll take here. This is CH2OH. So these carbon atoms are cyclized. Okay, then here the change is H is this side, OH is this side. This is OH, H, OH, OH, H. This is what? Beta D plus glucose. Beta D plus glucose. Understood? So this is alpha D plus, this is beta D plus. These two are what? Fischer structure. By comparing these structures, students, so here 1, 2, 3, 4, 5 carbon atoms are there. Okay, 5 carbon atoms are there, and the first and 6 are cyclized. Okay, so this is compared with the structure. This is compared with the structure iron. So by comparing this structure, we are going to write this glucose molecule that is called as Haber structure of glucose. Writing in this iron form. Okay, so let's start. So this is okay, so listen, this is carbon atom 1, 2, 3, 4, 5. Okay, so if you want to write alpha D plus glucose, students, if you know this Fischer structure, remember, whatever is written in the left hand side, that is in this plane, means this is H, this one is OH for first carbon atom. Second carbon atom, left hand side H, right hand side H. Third carbon, OH, H. Fourth carbon, H, OH. So fifth carbon atom, this is CH2OH, this is H. This is how to write the aromatic structure of this alpha D plus glucose. So this is called as alpha D plus pyranose. So remember, you just remember this structure. If you remember this, whatever the atoms which are written in the left hand side, they are represented above the line. Which are present in right hand side, that is represented below the line. Same for the same. Pyramid structure only, that is O, oxygen atom. So one. Okay, this is carbon atom 1, 2, 3, 4, 5. So carbon atom 1. So here OH is there, here H is there. Next, H, OH. Here OH, H. Here H, OH. Here CH2OH. Here H. This one is what? Beta D plus, not pyranose, it is glucopyranose. Glucopyranose. This is also glucopyranose. So these structures, both Haworth, this is Haworth structure and this Fischer structure, both are important for 2 marks. Remember, only in this chapter, the most oftenly asking question is write the Haworth structure of this and this. Listen, initially what they asked is write the Haworth structure of sucrose, write the Haworth structure of glucose, write the Haworth structure of maltose. Like disaccharides they are going to ask. But for your syllabus this year, they removed those related portions. So they removed those disaccharides and polysaccharides. That's why these are the main very important structures for your representation in this chapter. Okay, so always remember 5 marks one question is there, that is 2 plus 2 plus 1. 2 marks is also a structure. These 2 marks may be types or any other denaturation of proteins like that, or elucidation structures, their reactions, and 1 marks is the classification. Later I explain. Okay, so this is all about Fischer structure of glucose. So the next one important biomolecule is fructose. Okay, so the next one important biomolecule is the fructose. The molecular formula is C6H12O6. So students remember, same the fructose as the glucose, it has a elucidation structure. Molecular formula is C6H12O6. Again it is treated with HI. Then we came to know all the carbon atoms are in the straight chain. It is treated with hydroxylamine and also HCl. From that we came to know like glucose. So carbonyl group is present. Okay, it is treated with some other reagent which confirms presence of a ketonic group. Glucose is a aldohexose. This fructose is a ketohexose. Means it is a sugar having 6 carbon atom with a ketone functional group. But glucose is the sugar having 6 carbon atom with aldehydic functional group. Aldohexose, ketohexose. Okay, so the next one is fructose. From that we came to the molecular formula and all, and it completely converts into that word. Acid means carboxylic acid and all. We came to know of course 5 OH groups are there. Then it is treated with acidic anhydride. We came to know 5 OH groups are. Everything remains same. So I directly go to the structure because there is no elucidation structure of fructose for your syllabus. Okay, so now the structure follows. The 6 carbon atoms are here. 1, 2, 3, 4, 5, 6. Here is also CH2OH is there. Here is also CH2OH is there. Okay, and all are bonded. Here second carbon atom is having ketonic functional group, and the remaining follows as it is in the glucose. OH, H, H, H. This one is what? Alpha D. Okay, this one is glucose. So students, this one is cyclized. As I told you, this one is cyclized. There is no free organic group. There is no free ketonic group. 6. So this is CH2OH. Here it is CH2OH. So students, here carbon atom 1, 2, 3, 4, 5, 6. Here second carbon atom and this third carbon atom are involved in cyclization. Are you getting? So this one. Same happen here. Okay. If you want to take here, this completely, I'll just replace this one become OH. So students, remember, I'll just replace this molecule. I'll cut this part. I'll write this. Okay. Then CH2OH and OH is there. This is OH. This is CH2OH. If OH is remaining is same, H, OH, this is OH, H, same as it is. Okay. So students, if the OH group is right hand side, then it is alpha D minus fructose. Alpha D minus fructose. Remember. Next, same if I write 1, 2, 3, 4, 5 carbon atom. Of course, 6 are there. Then write this is bonded to what? 5th carbon atom. Here, if OH is left hand side, then it is beta D minus fructose. Okay, these are the Fisher structure. If add the structure which is involved in cyclization, only 1, 2, 3, 4 carbon atom with the oxygen. So listen, 1, 2, 3, 4 carbon atom. 1, 2, 3, 4 carbon atom with that ring. So that is representing furan. So fructose representing a furan structure. Now right here, oxygen atom. Okay. Now, so assume this is the first carbon atom. Okay, second, third, fourth, fifth. Okay, so according to this structure, okay, whichever is left hand side, here it is CH2OH, here it is OH. Next, OH, H, here H, OH. and this one is CH2OH H. Only this position decides whether it is alpha or beta. So here students, this is alpha D minus fructofuranase. Furanase then furanose. Alpha D plus fructofuranose. So if I write the aromatic structure here from this compound, I take oxygen. This is second carbon atom, third, fourth, fifth. Okay. So the thing here is here OH is here, CH2OH is here. Remaining is same. OH, H. This one is H, OH. This is CH2OH and H. What is the name of the compound? So I will write here beta D minus fructofuranose. That is the name of the compound. Alpha D minus fructofuranose. So students, what is this plus? What is this minus? Means plus represents the glucose D plus. It rotates the plane polarizer light towards right side. Minus represents it rotates the plane polarizer light towards left side. They are what? Optically active compounds. Understood? So this is all about fructose. The important is what? Hayward structure and Fischer structure. Very important. Both fructose as well as the glucose. 8 structures are there. Those 8 structures are very important. Alpha D plus or alpha D minus, then you can easily write beta because only this group is different in position, remaining all is same. Okay. So the next one is another important biomolecule, that is proteins. Okay, the next one is protein. So students, the proteins are the polypeptide long chain amino acid compounds. Okay, what are proteins? The amino acids involved in the peptide bond. Amino acid, listen. NH2 or COOH. These are what? Amino acids. So these involved in peptidization bond means NH2 COOH. Remember, 2 are there. So here, from here and here, H is there. H2O is removed. Not only that. So simply consider. Then it is NH2 or C double bond NH or CO. Just remember. So the CONH2 group is what? Peptide group. CONH group is called as what? Peptide group. Means the large number of amino acids, okay, are continually formed a peptide bond. Okay, so those are called as what? Proteins. Okay, long chain amino acids or long chain polypeptide bonds of amino acids are called as proteins. Okay, long chain polypeptides of amino acids are called as proteins. So students, they consist of nearly greater than 1000 to 10,000 polypeptide chains or polypeptide bonds, more than 1000 to 10,000. They are very bulkier molecules. Are you getting? So proteins, they are having very weight. Okay, macromolecules they say. Macro means very very big. Like how much? 10,000, higher than 10,000 polypeptide bonds are present in a simple protein. Okay, so in that next one is the classification of protein. So classification, very important for 1 marks. Remember this one. Classification of protein, 1 marks important. Then as 2 marks also. So first one, fibrous. Okay, first one, fibrous proteins. So students, these are, okay, these proteins are enumerate the polypeptide chains run parallel to each other, forms helix type or helix structure by hydrogen and disulfide. Hydrogen and disulfide bond. Okay, so the polypeptide chains run parallel to each other, forms helix structure by hydrogen and disulfide bond. Okay, so this structure is in the form of fiber. The structure is in the form of fiber. So it is called as fibrous protein. Means layer, 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 like a fibers. Okay. So example is keratin, which is present in hair, wool, etc. Another one is keratin. Another one is myosin, which is present in muscles. So students, I hope you see meat. In meat, a layer when it is boiled. So that is myosin. What is muscles? Okay. So those are what fibers. Here they are like fibers. Okay. So this is what a first type fibers like proteins. Okay. So students, they said what helix type structure means. As I told you, proteins consist of what? Amino acids. In that, of course, disulfide bonds are also there. So amino acid means this is one layer of amino acid. This type of structure they are gonna form. So here they have C double bond O and NH structure. These form bonds. Here C double bond O form bond. Here NH is here. Means NH they form bond like that. Okay. So by the hydrogen bond, by forming the hydrogen bond. So by forming hydrogen bond, okay, means the force of attraction between the highly electronegative atom and the hydrogen atom. So they are arranged in a helix type model that is in the form of fiber. Means, so I simply write, so these amino acids are arranged in this manner. They form a fiber-like structure, straight structure. Okay. Next, second one is globular protein. Globular proteins. What is globular proteins? Means the polypeptide bonds and polypeptide bonds. Okay, the polypeptides bonds in the protein are coiled around to form spherical structure like proteins. That is called as globular proteins. So here the example is, one is albumin, another one is insulin. I know you heard about this albumin and insulin. So students, how the structure is? Listen, these are coiled inside. Instead of arranging like this, they are coiled. Means like this. They form a spherical structure. This is what? A structure of a globular protein. Okay, so students, remember these proteins are water insoluble. Okay, these are water soluble. Okay, so you know, if air is there, I'm telling, if you keep this air inside the water so many days, so many years, no matter how many decades, it cannot be dissolved in water because it is insoluble in water. But if it insulin, you know that insulin for sugar patients, means diabetic patients, insulin is the main thing, means helps to digest glucose molecule. Okay, to check the sugar level. If the diabetic patients are there, their insulin level is very less, so then they have to take insulin. Why? Because this insulin is water soluble. It is not able to present in our body for a long time. Of course, it is easily soluble in water. You know, urine check they will do. So while the urine test, they'll check the sugar content. Means if insulin is more, then the sugar molecules, means glucose concentration in that urine is less. If insulin level is more, means the glucose concentration is more in the urine, then the production of insulin is less. Because of course it is water soluble, it is easily soluble in the water. This is water insoluble. Like that questions. Give an example for water soluble protein. Give an example for water insoluble protein. Like that. Give an example for fibrous protein. Give an example for globular protein. Like that they ask. Okay? This is all about proteins. So the next one is structure of proteins. Next one is structure of proteins. Okay, so what is the structure of proteins? Listen, there are four structures. One is primary structure, secondary structure, third tertiary structure, and quaternary structure. Primary, secondary, tertiary, quaternary. What is the structure in? Primary structure means the amino acids are arranged in this manner, in a series manner. This is primary structure. Okay, when these structures coil, okay, then they form structure like this. And if I want to say, the form means this type of structure. This is what? Secondary structure. Okay, so when this type of structures again they folded. If primary structure folded, then secondary structure will form. Fold like this. Okay, so if secondary structure folded, tertiary structure will form. Means this type of structures like this. They are folded. They form tertiary structures like this. And getting this type of structures again they folded, then they form tertiary structures. When these tertiary structures again they folded like this. Like these structures, of course, they are folded. Then this is quaternary structure. We cannot easily distinguish a particular structure in this quaternary structure. Primary, when primary structure folded, secondary, when secondary structure folded, tertiary, when tertiary structure folded, quaternary. Similarly, if quaternary structure is unfolded, primary, tertiary. If tertiary structure is unfolded, secondary. If secondary structure is unfolded, that is primary. These primary structure cannot break. Of course, if students, if they break, then the person will die. Understood? The proteins are the energy packets of our body. Okay. So listen here. The next one based on this is that is denaturation of proteins. Denaturation of proteins. So students, in this denaturation of proteins, what happens? Listen, if protein is subjected to physical change like temperature, pH, etc. is subjected to what? Physical change like temperature and also chemical change. And chemical change like pH means acid or base. Okay? The hydrogen bonds are disturbed and the structures starts unfolding from quaternary to primary structure. This process is called denaturation of protein. So students, example for this is coagulation of egg. Coagulation of egg. The second one is curdling of milk. Curdling of milk. So coagulation of egg means nothing but boiling milk. Okay. So listen, if egg boils, in that egg, the white color part is what? Albumin. Okay. If that albumin, when it is boiled at a greater temperature, means as I told you the physical change, temperature increases, then the albumin structure is quaternary. It is changes to tertiary. It is changes to secondary. Finally changes to primary. When it completely changes to primary structure, then it solidifies. And remember, this entire denaturation of protein is a non-spontaneous reaction. Means it is only forward reaction. It cannot be reversible. It is irreversible reaction or irreversible process. We can only break the structure. We cannot again form the structure. Once the egg boiled, it is boiled. Again, it cannot be converted to an original position. or original structure. Are you getting? So denature of protein is a one-side reaction, means irreversible reaction. Understand? So this is all about what? Proteins. The thing important here is what? What are proteins? Types of proteins, that is fibrous and globular. Fibrous are water insoluble, globular are water soluble. Example for fibrous is keratin and myosin. Example for globular is albumin and insulin. And this is the structure: primary, secondary, tertiary, quaternary. When they get folded in each other, primary changes to secondary, secondary changes to tertiary, tertiary changes to quaternary. When the denaturization of protein takes place, the structure changes from quaternary to tertiary, tertiary to secondary, secondary to primary. When the physical change like temperature and the chemical change like pH is applied. Okay, so that is called denaturation of protein. So this completes another one important biomolecule of the required for our body. So the last one for your syllabus is nucleic acid. Nucleic acids. So students, the nucleic acids are also called as hereditary carriers. Okay, right? Hereditary carriers. These nucleic acids are also called as hereditary carriers. Okay, so what are those nucleic acids consist of? So listen. Okay. So these are consist of proteins, amino acids as well as phosphoric group. Okay. So the simple thing, of course, these are also sugar moiety. The sugar moiety bonded to a base, that is nitrogen bases amino acids, and as well as phosphoric acid. So that together called as what? Nucleic acids. So students, nucleic acids are of two types. One is DNA, another one is RNA. Okay, so what is DNA? This is deoxyribonucleic acid. Deoxyribonucleic acid. This is ribonucleic acid. So from that you came to know the sugar moiety is a ribose sugar. Ribose sugar means the atom, okay, it consists of a 4 carbon atom. Okay, sorry, 5 carbon atom. Okay, listen here. I'll write the structure DNA here. This is 1, 2, 3, 4 carbon atom. Okay, of course 1, 2, 3, 4 carbon atom. Another one is here. Total 5 carbon atom. If 5 carbon atom sugar is there, that is what called as ribo sugar with a 5 carbon atom. Okay, pentose sugar indirect. Okay, so this one is H, this one is H, this one is OH, this one is H, this one is OH. Okay, wait. Okay, initially I will write complete structure. Later I will show you. This is OH, H. This is alpha, sorry. OH is beta, D, ribose. Beta, D, this is name. Students, this is not the structure of DNA still. Wait until I write the both the structures. This is OH, this is H, this is CHOH. This is CH2OH, H. So this is beta D ribose. Okay, so listen. Here in these molecules, the only thing change here at the second carbon atom. This is first, this is second. Oxygen atom is removed. Are you getting? So your oxygen atom is there. Our beta D ribose. Here at the second carbon atom, oxygen is removed. It is H and H. Beta D2 deoxyribose. Deoxy means oxygen is removed. This is DNA structure. This is RNA structure. Okay. So students, this structure of nucleic acid. This is DNA. This is RNA. Nucleic acids is nothing but consists both DNA and RNA. Okay, what is the structure of this nucleic acid? What it consists of? So nucleic acid consists of two parts. One is nucleoside. Another one is nucleotide. Right? One is nucleoside. Another one is nucleotide. Means the structure of nucleic acid consists of nucleoside form as well as nucleotide. So what is nucleoside means? For the first carbon atom, this is the first carbon atom. If first carbon atom is bonded to amino acid base or amino base. First carbon is bonded to amino acid base, then it is said to be your nucleoside. What is nucleotide means? This carbon. Which one it is? The fifth carbon atom. Okay. So here, fifth carbon is bonded to phosphoric acid group along with nitrogen base. Along with in bonded to, you take here nitrogen base. Nucleoside and nucleotide means nucleotide is always present along with nucleoside. But nucleoside cannot be with nucleotide. Means alone nucleoside is formed. Means here I take this is a okay. If this OH group is the instead of this OH group, I am writing here DNA. I am considering base is there. Then this is what nucleoside. Only this type of structure. This DNA is a C1 carbon atom base is there, that it is nucleoside. So and at the same time, this CH2 O and P double bond O. Phosphoric acid is there. Then together this molecule is nucleotide. Here also, in this ribose, if it is bonded to base, then it is nucleoside. Along with base, if it is bonded to phosphoric acid also, then it is nucleotide. Are you getting? Only RNA, only DNA cannot be possible, cannot be present like this. They are not in the form of a ribose sugar. They are in the form of a nucleoside and nucleotide. What is nucleoside? For the first carbon atom in the RNA or DNA, if nitrogen base is bonded, then it is nucleoside. Along with the nitrogen base, the fifth carbon is bonded to phosphoric acid, then it is nucleotide. Very important. Nucleoside and the nucleotide. One mix. Okay, and this is deoxyribose and that is ribose and deoxyribose. Okay, so the next one, students, the very important thing is what are the nitrogen bases present in DNA and RNA. So there are 4 nitrogen bases are present in DNA and RNA. So in DNA, The nitrogen base present in DNA is adenine, represented by capital A, guanine, represented by capital G, cytosine, represented by C, and thymine, represented by capital T. Okay, these are the nitrogen bases present in DNA. So the nitrogen bases present in RNA is adenine, no doubt. Of course, guanine too. Next, cytosine. First three are same. The last one is instead of thymine, uracil is present in RNA. Are you getting? So the one marks very important question. Name any one nitrogen base which is present both in RNA and DNA. Among these three, enumerate anyone. Enumerate adenine, guanine, and cytosine, because these three are common nitrogen bases in both DNA as well as RNA. If particularly they ask, name the amino base or amino acid or a nitrogen base which is present only in DNA, not in RNA, that is thymine. Okay, next, name the nitrogen base which is present only in RNA, not in DNA, that is uracil. Except last one, all the three amino acids, nitrogen bases are commonly present in both RNA and DNA. So these decides the hereditary of the children. Okay, hereditary character of the human beings. Okay, so DNA model is like this. Double helix model. Here the nitrogen bases are going to be coding. Okay, in this arrangement of these nitrogen bases, there is a difference that decides the hereditary character of the human beings or the living organisms. Okay, this is all about the repetation class of the biomolecules. This completes the repetation class of biomolecules. And remember, only these three topics are covered. Very easy and compulsory 5 marks question is there. So do practice on this chapter, because 5 marks it carries and it is very 6 marks it carries, and it is very easy because the most of the concepts are removed from this topic. And getting in this chapter, all are removed. Vitamins are removed, enzymes are removed, amino acids are removed, hormones are removed. Only these three are there. Among these three, 6 marks questions they will ask. Okay, so keep on practice the very easy. Thank you.